So it's a great pleasure to have Peter Bankhead here today, who is a senior lecturer at the Center for Genomic and Experimental Medicine in Edinburgh, and he's the developer of one of very widely used systems called QuPath. And we're very happy he's here today to give us an update on what he's doing and how his system is being used. Peter, over to you.
Thank you very much. Thanks to the organisers and to you, Florian, for the invitation to speak here. At the risk of testing that hypothesis too early, has anyone used QuPath here? Some people. Do people here work with images very much at all? With other software then? Okay, good to know. That's fine. So I'm going to talk to you about QuPath, and I'm going to show it to you a bit during the presentation today. I want to begin, though. So my background is in computer science, as you'll see in a moment. But I've always ended up working with biologists, and most recently pathologists. And typically, whenever I'm working on a project, the image analysis is really the big black box, where the images that go in, and then some kind of stuff happens, and then the results come out. And the people I work with are mostly interested in what goes in and what comes out, and then my responsibility is in the bit in the middle where the stuff happens. And that's something where I don't really want all the responsibility on that, because as the computer scientist, I'm not the one who necessarily is going to really understand how to interpret the results. So what I want to focus on is try to make the black box as transparent as possible and to actually share what's going on in there and make it accessible to everybody. And so the challenge for me and for people like me is to try and come up with better and more transparent algorithms that make this possible. So bioimage analysis involves different groups of people. We have the ones who write the software and algorithms, and they're the ones that I can relate to. And then the ones who use the software and algorithms and complain to the ones on the other side about the ones that they've created. And so we have to all somehow work together with our different kind of expertise, which can lead to communication difficulties. But I think that in order to be able to work together, images are quite a good topic because they're so visual. And I think the visualizations and the software that can be developed are what allows us to work in this cross-disciplinary way and to be able to draw out the right kind of information from the images that we should. So as I mentioned, my background is in bioimage analysis. I did a PhD at Queen's University in Belfast, focusing on calcium signals and retinal blood vessels. As a sort of a side project, I developed this retinal blood vessel analysis software, which ended up being more popular than anything else I did during the project. And then after that, I moved to Heidelberg University, where I worked at a core microscopy facility. And there, I started off trying to treat every project as a research project and spent a long time developing an algorithm. And I realized that wasn't going to make the people using the microscopes in the facility very happy, because what we needed is faster solutions that answered the precise questions that they wanted to address. And this is what really pushed me slightly away from pure algorithm development and a bit more into the open source software and using things like Fiji and so on to be able to try and create a customized analysis pipeline for specific applications as rapidly as possible. So while I was in Heidelberg, one of the things that I discovered is that lots of people were coming with similar sorts of questions. And I thought that I could try and answer all these questions individually, but I'd probably give better answers and help more people if I can try and explain these concepts in a broader way. And so I wrote this image analysis handbook, thinking that I could probably write about 20 pages and that would describe everything that you would need to know, but it ended up being a bit longer than that, either because there was more to know or I was a lot wordier than I intended to be. So this is freely available online as well, both as a PDF and as a Git book. And so this is really my attempt to try and explain the concepts of bioimage analysis to people who might not have the time or the interest in delving into the really heavy textbooks in the field. And it tries to really focus on the practical skills and techniques that I used every day in the microscopy facility. And through all of this, I've come to the conclusion that when it comes to image analysis, we often end up measuring what we can measure and not necessarily exactly what we want to measure. And so it's very often the case that maybe we've got something really complex in the image that we want to be able to detect and quantify. even just detecting it is so incredibly difficult, we end up measuring a stained area, something like that, something simpler that we can do as opposed to what we necessarily want to do. And if we do that, it's important at least to know that we haven't exactly measured what we wanted to measure and not just assume that it is equivalent, because that may not be the case. But I also think that we need more software tools, and new software tools that allow us to answer biological questions more precisely. So it's not the case that we always rely maybe on the same one or two software platforms that might be very general and incredibly useful and important, but it also helps to have some slightly more specialist software platforms as well. But I think open source has a unique role to play, because if you develop your software open source, then everybody has access to it. They can begin to explore new ideas, develop new applications, algorithms, customize it in new ways that certainly I would never have thought of, or the developer would never have thought of. It makes it possible to run the analysis that's required for maybe new, completely novel or niche applications where there's no incentive to build a commercial application, but an open source tool could be customized for it. And for research, open source is essential for transparency and reproducibility. So, having worked in Heidelberg in the microscopy facilities, developed some of these opinions around it, I came back to Belfast at the end of 2012 as a postdoc working in digital pathology. And initially what I wanted to do is to use these open source tools that I was familiar with, like in particular VG and ImageJ. But the trouble with digital pathology is that none of these really worked. So prior to that, I worked a lot with fluorescence microscopy images. I came up with ideas for how to work with them. They shouldn't be JPEGs. You need to store raw data, uncompressed, all that kind of stuff. I worked in the field of digital pathology then and discovered that the images were horrendous compared to everything that I wanted an image to be. So these are huge. So you could have a single slide. In this case, uncompressed, it's about 60 gigabytes. You can see there's a lot of white space, so you wouldn't necessarily think that it's using the 60 gigabytes quite as it would need to. But if you were to just crop out the tissue region and look at it at full resolution, it would still be about 10 to 15 gigabytes of data. Often the data is JPEG compressed, and it's not terribly good for quantification. But this is the kind of data that is commonly worked with in pathology. And if you've got a computer with maybe 8, 16, 32 gigabytes of RAM and you have an image that's 60 gigabytes, then clearly you can't open the entire image in one go because you're going to run out of memory. But in a way which it can work in this sort of Google Maps type way, inside the image file in pathology, the same image data is stored at multiple resolutions and divided into these little square tiles. And so you need software which is able to perform the visualization and analysis by going into these pyramidal images and then pulling out only the tiles for the pyramid resolution that's relevant at that time, rather than read the entire thing into memory. And that's one of the primary reasons why the existing open source software, whenever I came to work in this field, just wasn't really suitable for pathology images. So as I started my postdoc, I spent the first 2 years trying to use existing tools. I wrote some ImageJ plugins, a really convoluted Python-based parallel processing framework. They were horrendous to use. No one else ever used them apart from me, and I wouldn't know how to use them now if I saw them again. But again, this was similar to when I started in Heidelberg. I thought, treat it as a research project, come up with a new algorithm, a new way of doing it. But I forgot to pay attention again to really the usability of it. And at the end of these 2 years, I'd got some stuff that potentially could be publishable and might have looked like it was useful, but genuinely never really would have been useful because it was so convoluted and cumbersome to use that it wouldn't be applied in practice. So then, for the final 2 years of my postdoc, I focused on what was the platform that I would like to be developing with, or what is the software I would like to be using for digital pathology, and that's what then became QuPath. So I set about creating that. The software that I'd tried to avoid creating for 2 years in the false belief that it would be faster to try and adapt what was there before eventually realizing that it was actually going to be faster to try and make something that was designed to do what it was supposed to do. And so you might have seen a video like this before. I'm not going to show the whole thing, but this is QuPath as it was when I was just left Queen's University, which is when the last stable release was made. It shows the analysis of tissue microarray. The image is about 40 gigabytes, and the video is running in double speed. But if I was to run it entirely, you would see that within 5 minutes or 2.5 minutes double speed, you're able to detect hundreds of thousands of cells, classify them as positive and negative, and score this tissue microarray in about 5 minutes, which is pretty fast for the kind of data that we have here. But I'll come back to show some of the tools in more detail later. But one of the points that I try to make whenever I'm describing QuPath is that it's a software platform, it's not an algorithm. So I tried to step away from developing one algorithm for one purpose into trying to find the common themes of how could I solve lots of problems as efficiently as possible in one kind of platform. And that's really where QuPath came in. That's the idea behind it. So the goal in developing it was to make quantitative analysis more rapid and reproducible, and in particular to deal with this case of pathology images, so these really large pyramidal whole slide images for which existing open source solutions weren't suitable. In doing that, it could be the case that this would also result in some tools that were useful again in fluorescence microscopy or useful for other imaging modalities, as we'll see later, but the goal was really to come up with something that met an unaddressed need. I wanted it to be open source, and I wanted it to be suitable for pathologists and biologists to use, so it should be user-friendly, but it should also be suitable for image analysts and algorithm developers, because I didn't want others to have to spend 2 years like I did making something that wasn't very usable. I wanted to be able to streamline the process of developing good algorithms and working efficiently in research. So here then is the publication of QuPath then that came out soon after I left Queen's. Screenshots show some of the diverse applications of the software, but I'll show it to you in a moment later. In this publication, we were looking at scoring breast cancer biomarkers. So I should say, if you're not familiar with tissue microarrays, each one of these circles is a tissue sample from a different patient. In this publication, they were primarily from colorectal cancer patients. And we were looking at different biomarkers, so each of them stained for different biomarker, and we were showing that we could perform the analysis of these biomarkers in QuPath across thousands of patient samples from about 600 to 800 patients. And we could perform this analysis very efficiently and reproducibly. But in addition to these tissue microarrays, these small circles, we were also showed within this publication that we could look at something like the tumor stromal ratio in a larger tissue sample using a kind of a texture analysis. And so again, it's focused on the fact it's a platform as opposed to one algorithm for one particular thing. You can adapt it to these different applications. A second publication from my time at Queen's was looking at breast cancer biomarkers. Again, it was still primarily focused on tissue microarrays because that's what I had mostly at the time. Looking at ER, PR, Ki67, HER2, and P53. And in this case, unlike in the colorectal cancer case, we had pathologist scores and we had patient outcome data. And so we could see that we could score these biomarkers, and it showed all the associations with overall survival that we were expecting to see, but it also had very high agreement with the pathologist's scores. So that gave us some confidence that it was doing what it should do, but it's very hard to validate these things, particularly whenever it's so customizable. And so all of this is just kind of really one angle on a validation of it. It doesn't fully prove the software for any one specific application. You kind of need to think about how you validate for each application that you might want to apply it for. But because it's open source, so the last stable version was released on my last day at Queen's University as a postdoc. Since then, it's had more than 40,000 downloads. If you count some of the other versions since then, it's well over 50,000. And it started to be used really all over the world. I try and keep up with the publications that are using it. Please do cite it. It makes it easier for me to find. And then, so it's starting to become very widely used, particularly in pathology, but there's a lot more that it can or could do over the next year or two. There have also been some workshops in Europe and North America, but what I found through creating this is it's not really just about the software. There's also a user community, and the user community often do much more exciting things with it than I ever have, and think of new ways to apply it. But in order to support that, there needs to be a community that exists online, and it involves creating the documentation, tutorials, and teaching materials as well. So it's much more, so the initial publications behind it are just a very, very small part. The overwhelming majority of the work is in developing, supporting the software. But one of the publications that particularly caught my eye was one from Professor David Rim's lab at Yale, where they independently looked at it, and I didn't know they were doing this until after the work was done, and they showed that in comparison with some commercial platforms, which are rather more expensive, they could score Ki-67 in breast cancer using QuPath. Their conclusion, was that there was outstanding reproducibility within between the image analysis platforms, including with QuPath. And they were essentially indistinguishable with respect to prediction of breast cancer patient outcome. So as a lot of you may be familiar, the standardization of Ki-67 scoring in breast cancer is troubling, or it's a difficult thing to do if pathologists are assessing it by eye. And so image analysis potentially offers a way forward there. So immediately after I left Queen's, I was in industry for a year, so I wasn't actively working on QuPath. I wasn't really able to. But in September then last year, I joined the University of Edinburgh to continue QuPath. So I moved from Belfast to Edinburgh. And since then, I continue to be the only person working on it, but hopefully that's going to change in the next month or so. And I've been making new releases of it. I find it is harder to make new releases of the software when there's thousands of people using it, as opposed to whenever the only user was me, but it is more interesting as well. So I started to make these new milestone releases where I try and make new functionality available to show the direction of the project, and also to make potentially help some studies that might be happening in the meantime. But it's still kind of in a state of flux. It's not yet a stable new release. And so if you want the stability, you probably want to go back to the version before. That's linked to in the website. But over the last few months, I've been making these milestone releases to show the direction of the project and to get feedback and try and get other people involved in it. And that's what I'll show you in a moment. So the guiding principles then behind this is, I think that the hard part of image analysis should be defining the question, and it shouldn't be trying to wrestle the results out of the software, as can often be the case. I still think there is an awful lot of trying to wrestle your results out of QuPath if you're using it, and so there's still more to do here, but at least the goal is to try and to make it as efficient as possible to answer the exact question that you want using the image analysis software. Image analysis should be verifiable, so it shouldn't be the case that you put an image in, you get a number out, and then assume that that number relates to what it should do. You should be able to go and be able to visualize what has been done, what has been detected, quantified, and so on. So I think the visualization tools are really important, and the interactivity is important. You may want to run your analysis in batch across 100 or 1,000 images, and you should be able to do that, but you should also be able to go back and really check what has been analyzed. But I do think that in research, there's so many different problems that we're working on. And with image analysis, almost everything needs a solution that's in some way customized. But if you can come up with software and algorithms that sort of span across different applications and implement them in a reasonable way, then they can have a big impact, because then lots of people can use them. And they can use them in new and inventive ways, and they can do much more with them. So my main area of focus when I was initially developing QuPath is to create the platform that would give a whole slide viewing and annotation software, which didn't really exist in an open source way at that time. I focused a lot on cell detection and superpixels, which are basically like these areas where you might want to do texture analysis, like for this tumor stromal analysis, and how you represent all of this in a kind of a data model. So that's what I focused on. I'm going to jump out of the presentation, and I'm going to try and show you this in practice. I'm going to show it to you running it in debug mode, the version that I was still changing on the flight on the way over. So please, I do expect that at some point this will let me down, but it's more interesting to use this as opposed to the one from a couple of years ago. So this is the beyond the latest milestone version. So this is the code that I'm currently working on myself. Okay. So here we have QuPath, big black screen at the moment. Here we have a list of images which I've categorized in a project according to where they come from. If you've seen me give QuPath demos before, I apologize for showing this image again. I really wish there was more public data sets of whole slide images, but I'll show some different images in a moment. But this is a useful one. It's key 67, and it's a useful one for showing the concepts. So if I zoom in, you can see this is the high resolution image. And if I zoom out, you can see that this red area is how the field of view corresponds to that part of the slide. And so this is, as I described, QuPath is then going in and pulling out the part of the image that it needs to at any particular time. If you're working with an image like this, what I'm going to want to do for the analysis is get the percentage of tumor cells which are brown. So I need to be able to identify the cells, classify them as tumor and non-tumor, and then classify them as positive and negative, so that's what we'll see. But I said that I think visualization is important. One of the ways in which it can help is through even being able to digitally separate the stains. So you can see quite clearly what's blue and brown there, but there is a computational technique called color decomposition. And if I just press the shortcut key, I can apply it and separate the stains, or I can choose it from a list if I want. So there's lots of shortcut keys because I primarily created QuPath not to create the software platform because I needed the tools myself. Okay. And actually, if I remember, I keep forgetting to do this, you can script QuPath. So I have written a script which will show you the shortcut keys that I press because I get carried away with the shortcut keys. So the fact that you can script it has become one of the most important features of this. So if there's something that it doesn't do, you can potentially script to get it to do it. Okay. Right. So what we want to do here is to be able to identify the cells and to count them, which sounds simple enough, but as you zoom in, you see that it's, from an image analysis point of view, it's harder than it might initially seem, because the cells can be packed together, different shapes, and you need something that works quite generally. And so one of the algorithms I developed in QuPath is for cell detection. It is intended to be very general, so you can apply this type of image, you can apply it to H&E, you can apply it to fluorescence and so on, but you might potentially want a more optimized algorithm in the future. I will show it then in action, and I will stop going to this menu because I always get lost in the menus, and I'll show you the alternative to that. Okay, so here's the cell detection, lots of parameters to choose. If I run, then this is the results. I can press F for fill, and it will show you what it has detected. And I can press D to show and hide the detections. And so you shouldn't be too convinced by this. You can see that it's done some pretty nasty fragmentation in here, and so it's made some mistakes. But I mentioned that you can view these digitally separated stains, and these relate to some of the parameters up here. So you can see that the stain separation isn't maybe entirely clean in this image. And so depending upon the kind of staining that you have, you might find that you need to use different parameters. So I'm going to switch, and I'm going to use all of the staining that we have without trying to separate them for detection. And if I run it, then I think you'll find there's a detection which is a bit more convincing. May want to adjust some of these other parameters if necessary. And if you put the mouse over, there's help texts that describe them and YouTube videos that explain it online as well. Okay, so once we're relatively happy with the settings, I will delete that then. So I just chose a small region to get started. And then I'll choose a larger region. So normally I run a lower resolution. So I'm just going to, I thought now's probably the time to try a higher resolution, see what happens. Okay, so it's breaking up the image into different regions, running the cell detection across each of the regions, and then trying to resolve the overlaps a little bit. So what we have is within this region, we have about 40,000 cells that is detected. You can see that it starts off by detecting the nucleus, and then it expands this out. And you can control the expansion if you don't want it to go so far, until it goes to a fixed distance or crashes into the neighboring expanding nucleus. If I double click on a cell, you can see that it's made about 30 to 40 different measurements for that cell. And all of this is represented in the image. And I can adjust the opacity. I can adjust how they're displayed to be able to see it as clearly as possible. But if you've got 30 to 40 different measurements and 40,000 cells, then it could help to be able to see them within context. So we have then this additional visualization, which allows us to get basically heat maps of any particular measurement. And so the one that's quite informative for distinguishing between tumor and non-tumor cells is the nucleus to cell area ratio. So there's other measurements of staining intensity in the different cell compartments, size and area and so on. But the nucleus to cell area ratio is particularly useful. I can adjust the heat map if I want. And so you can see that these denser areas, which I'm not the one qualified to say this, but I'm going to say tumor cells here, do end up with higher values, as you can see through the heat map, for the nucleus to cell area ratio. With some of the others, there may be some kind of connection, but it's a little bit less distinct. But in itself, that's probably not going to be enough to be able to distinguish between different cell types. So what we may want to do then is to be able to train up a machine learning classifier, which we'll do in a moment. But we might want to give it a bit of extra information as well, because if you know the cell of one, if you know the type of one cell, then very often the neighbors may be the same cell type, or at least it's relevant what the neighbors are. So I'm going to go to the shortcut. I'm going to add in some additional features for every cell. So we've got our 40 measurements or so. And now for each cell, I have supplemented these with smooth measurements. So essentially, I've calculated a weighted sum of the corresponding measurements for neighbors and calculated the local cell density and so on as well. We could calculate different additional features like textures or whatever else we might need, but this is a simple and quite fast method to get. So now, with this, initially, I should have mentioned it, you see some of our cells are red, some are blue. This is based upon what QuPath thinks is positive or negative when we run the detection. But we don't need to worry about this too much because we're going to override it in a moment. But currently, we only have the number of positive and negative cells and the number of cells within this region. But because we have all of these additional features, I can then draw an example here. And I'm going to risk giving bad cell classifications, but I'm going to take a chance that these are going to be stroma, these are going to be tumor. And we can ask QuPath to create a detection classifier, tell it to auto-update. And then it will learn from these features that we've got, and then classify all the cells across the image. We can then now make the decision to stain based upon the nucleus staining, to score based upon the nucleus staining to say a cell is positive or negative. So now we have blue is positive or negative in tumor, red is positive in tumor, and then green, light green or dark green is positive, negative, and non-tumor. But because this classifier is trained very quickly, we are able to go into other regions, give it more examples, and then interactively train it however we need to if we see that it's making mistakes. And as we do it, we can see that our summary measurements are automatically calculated to give us the percentage positive in the different, whether it's going to be tumor or stroma. So if we want to, we can adjust the features that go into the classifier. We can adjust the threshold. And you should see that if we increase or decrease the positive and negative threshold, then the results that we get here will change. We can save the classifier and we can apply it again if we need to. All these steps are logged, and so we can go back and we can open the cell detection with the settings that we had before. But we can also create a script and then run it across the project if we want. And so this is what helps with reproducibility. So the goal here isn't really to come up with an incredibly general Q67 scoring algorithm. It's to be able to come up with a customized one for a specific data set, for specific staining, scanner, and so on. But through the insights on these kind of smaller scale studies, that could then feed into creating a much more generalized algorithm in the future. And so it's the interactivity and the speed with which you can create the customized algorithm in QuPath is one of the biggest and most important features. And I did mention then about this object hierarchy idea. So you don't need to see it at the moment, but QuPath will then keep a record of where the annotations are inside one another. And so that's what it means by hierarchy. I can move it and it will store these relationships. And so this then allows it to represent something like tissue structure at different levels, where you can have this kind of hierarchical representation. So at the minute we just have a region, we have cells within it, but we might want to go a bit deeper and to represent something more complex. Okay. Feel free to ask questions at any moment as well. So that's really QuPath. Some slight updates there, like the lookup tables for the color map, but that's essentially QuPath's features as they were whenever I left Queen's. So what I'm now working on is more on the pixel classification side. So you don't necessarily have to generate cells and then classify them, but try and take some of these concepts and make them a lot more generalizable, applied directly to the pixels in the image. Multiplex analysis for fluorescence or imaging mass cytometry images, where you might have a very large number of color channels, essentially, for a large number of markers. And then how do you interrogate the data, look at spatial distributions and so on. Okay, oops, I think I have one more slide on that. And so my goal is to try and make, with the pixel classification, is to try and make it intuitive to go from an image, to applying different types of classifier to identify regions. So if I switch back again to QuPath, and if I go to a very simple image, if I go over here, you can see the entire image is about 26 gigabytes or thereabouts. And let's say we want to be able to identify the brown stained area within this. So don't worry too much about the classifications that I give it. I showed then the rectangle tool in QuPath, but there's actually far more powerful annotation tools. So here's a magic wand, tells me to select a region to begin. I'll choose another region here. I will go into the new pixel classifier. Again, I'll say live prediction. And similarly to in the case with the cell classifier, it will then begin to train up a classifier across the entire image. This is now based purely upon the pixel information and not based upon detecting anything first. I can say that I want to eliminate the white space, so I'll give it an ignore class and it'll exclude it. But the thing with these images is that sometimes what you want to measure is at high resolution, sometimes at a low resolution, so we can adjust between that if we need to. And QuPath will then adapt to train up a new classifier depending upon the resolution that you need it at. And it will give you then for each region, for any region that you get, the percentage of each class within that region. If, mm-hmm. Yeah, you can. So you can add whatever classes you want in here. There is no built-in limit to the number of classes, except, well, as of the last few days, there might be but it's like maybe 255 if it stores in an 8-bit image, so thereabouts. I suspect the issue is going to be the features, calculating the features that enable you to distinguish those classes. But yeah, there's other alternatives to that as well. Let's see. Whenever you have this, you can then, for example, generate annotations from whatever you've classified. And so, for example, here now we've used this to identify the region that we might want to apply further analysis within. But as I said, my interest is in finding what are the common themes that we might want to solve. And so we can take the same pixel classifier and apply it then for quite a different sort of application. So here I'm moving into a fluorescence image. And so this is, I gave a workshop at the La Jolla Institute, and they generated a lot of really nice data sets. So this is one of theirs. So here we have a fluorescence image. I can turn channels on and off. And let's say I'm going to want to go to the nucleus channel. I'll view it grayscale. And let's say I want to come up with a cell detection if I'm not happy with QuPath. So I can apply this magic wand, which will adapt to whatever resolution I'm at. So if I want to create a small region, I just zoom in. I'm going to say this is nucleus. I'm going to say this is nucleus. And I want to give it some background. But I don't want to have to draw too carefully. So I can hold down a shortcut key that prevents this overlapping with something else. I'm going to say I want it to ignore this. And then I'll train up a new pixel classifier. It is horrendously bad at the start because I need to run at a high resolution. But now it starts to look more sensible. But let's say that, okay, I want to adapt the features. So I only give it the nucleus channel, and I want to give different kinds of features to the classifier. I can choose this. If I want to show them, I can press the show button. And this allows me to visualize the information that QuPath is basing its classification decision on. And you can see that maybe there's a chance that it will be able to identify the boundaries between nuclei as well. So a new option added in the last day or two allows us to be able to train a classifier that also attempts to learn the separation between cells. And we can output that, say, as probability. Oops. And so it may not be too convincing yet, but we can also change the type of classifier that we're training it until we come up with something that actually is more capable of the job. Okay. Okay, and all of these options are in there for customization if you need to try and come up with something that works for that application. So from my point of view, it's the same fundamental techniques, but just applied to different types of images and trying to come up with them and implementing them in a general way so that you have the options to customize that you need to be able to adapt it for whatever you need to apply it to. So this is still a work in progress, but it's not applied only to pathology. And so I think I need to duplicate this to make the video run. And I am going to need to close that little window. So my partner is an immunologist, and so I always need to make it work on her images as well. And so she's working with fluorescence images. Here we're looking at quantifying myelination. And so this is a Z stack from a confocal microscope. And unfortunately, my video is now starting to play. Again, QuPath's pixel classifier is allowing us to be able to identify these regions. And this time using the Z stack and calculating even features in 3D now. So what I haven't said anything about is deep learning. So this is using conventional machine learning techniques, random forests, but also neural networks are different types of classifier that are available. I didn't speak about deep learning, but that is really the main thing. It's certainly in pathology image analysis, but much more broadly as well at the moment. And one of the things that I'm trying to do with creating these tools within QuPath is to make them extend and apply to deep learning as well. So I know that one of the ways QuPath is widely used is purely as an annotation tool, both in academia and industry, to create annotations of regions and then train up a deep learning model afterwards. So what I've done within QuPath is I've spent a lot of time in the annotation tools. I've written some scripts that allow me to connect with, say, Jupyter Notebook, so I can link up with Python and so on. I create these tools kind of that I might find useful for wider applications. But I'm also looking to develop a kind of a deep learning segmentation workflow within QuPath and other deep learning pipelines within QuPath. So as an example, which I haven't presented this before, but this shows in general how it works, we can use all of these annotation tools that I showed, where you've got all of the different tricks and magic wands and so on to help you annotate really accurately, really quickly, and even rely on a conventional machine learning classifier to bootstrap your annotations and help you make them sooner. I said you can script QuPath, so you can export these in whatever training format you need for your deep learning framework. So then you train up your deep learning model, say using TensorFlow or PyTorch, and then you can visualize this within QuPath again. So I think I have an example of that. I've closed down QuPath, so I'm going to have to open it again. So the aim would be that if you're, say, somebody who's working in image analysis, wanting to apply deep learning, QuPath can help you do your research more quickly without you having to, see it as QuPath develop your algorithm for you. It helps you develop the algorithm you want to develop, and it helps you to share it in a user-friendly way with others. So here, if I open up this script, so it's still a kind of a prototyping proof of concept sort of stage, but this, I hope, yeah, it's going to end up crashing. For the first time, I tried that today. But yes, you might have got a glimpse there whenever it opened up, where it was able to show the beginning of the deep learning classification through the pixel classification framework. I think I know what I broke there, but that's going to be a task for another day. Yep.
Just two very quick questions. I didn't quite catch which deep learning framework you were saying you were using, because obviously you managed to get this working in some which.
So it shouldn't necessarily matter. So the way in which I was running this and the way in which you can see is giving me a little bit of trouble is using OpenCV, which can read in pre-trained models from a variety of different sources. So this one I used TensorFlow. There are some issues with making sure that your model is compatible. Wow, yeah, I really destroyed that in the last day. Excellent. Yeah, so it has to be something that is compatible with OpenCV. But I've implemented it in such a way that you could also use something totally different. So lots of this is defined with interfaces in the idea that somebody might want to come up with an extension to QuPath that integrates with something very precisely. And actually, I would like to do that as well myself, because I think that the OpenCV one has some limitations. Crashing catastrophically is one of them, but I can't say that's their fault and not mine. But yeah, so that's the...
Yeah, and the other one was just, do you have, so obviously, like, when you're coding, it's useful to have examples and things. How many of the examples do you have?
So in terms of the deep learning, not really any, because as you can see, it's very much a work in progress. It did generate this image. It has worked in the past. I have a blog where I post scripts for things which are a bit more established. But yeah, I try to get the balance between documenting things, but also then I might change them. And what I'm finding is I change things. I spend a lot of time trying to answer questions about how to update your scripts to make them work. And it's my own fault for changing things, but the reason for changing it is to try and make it easier in the future, which makes me a little bit more reluctant to put things on whenever they're in quite such a fragile state as this. But this is, yeah, where it's going. Yeah.
Hello? Okay, in digital pathology, we often found difficult to detect very quickly staying cells and cells which are closely located. Mm-hmm. So in your software, are you applying specific method analysis?
So I don't think there's a good solution to that in QuPath at the moment, but I would like QuPath to be able to support the good solution that somebody comes up with, which could be you. One of the other things that I've been doing recently is making it possible for QuPath to transform an image as it's being displayed. So it could apply like a color normalization, something like that, all calculated dynamically as you view the image. So it could be the case that you try and normalize the image to deal with the problem that way. Or it could be the case that you train up a model which is hopefully robust to those variations and then apply that. None of that is currently within QuPath, but it should support either solution whenever somebody develops a solution.
When you detect the cells, initially you are segmenting the boundary of the cells.
Mm-hmm. The nuclei, yeah.
Yeah. So as I saw a little bit, the boundary is bigger than the cell area. Mm-hmm. So how reliable is the features you extract from this, especially the shape features?
Do you mean of the nucleus or of the entire cell whenever it's expanded out?
the entire cell?
I would say not reliable, and that's why multiplex analysis is one of the things I really want to work on, because I think that's a big issue. And that's also one of the goals of trying to get the deep learning support within QuPath and to have the ability to have cell segmentation algorithms that work for that kind of data. Because I think that can be a big source of error, and I don't have I think it's a difficult research problem to solve in a way, certain, even for one image set it is, but to generalize across different types of staining, it becomes incredibly hard. But I think it's a really important one. And so, currently in QuPath you have a distance-based expansion. You know it's going to make errors. What you can do is you can constrain it to maybe only expand 1 or 2 microns, so you know it's going to be close to it and it's less likely to assign a density from a neighboring cell, but it's not entirely satisfactory. But it is reassuring for me if I interpret that right, that you agree that's a problem, because I really think it's a problem. And I am happy to know that that opinion is shared. Okay. Which brings us on then to, there's more to QuPath than machine learning. So there's, for instance, microscopy image support. So Yvonne, my partner again, so this is one of her images. Even stuff like manually counting double positive cells I find can be difficult because it depends upon the brightness contrast of the image that you've got. So that's why we have these shortcuts that allow you to turn channels on or off. So if you're doing cell counting currently with Fiji, and if you're happy with it, then stick with it. But if you find this to be a problem, this is the reason why some of this exists within QuPath, because I was trying to help with even like manual cell counting with all these shortcuts. More recently, though, I've introduced this channel viewer that allows you to visualize all the channels simultaneously. And I'll show in a moment, but I'll try and show it live as well. This should expand to quite a lot of channels. So if I open up the channel viewer, so this is a multiplex ion beam image. 44 different channels. You can visualize them all simultaneously within QuPath, and you can adjust, if you want to, the magnification. And so if you want to come up with your fantastic, the more accurate cell segmentation method, then this might be of help to you, because you have if you need to create ground truth, you have, for example, the magic wand tools, all that kind of stuff. But you can then visualize, as you annotate, all of the channels simultaneously, and you can know really what has been detected. If I manage to get the cell, if I manage to get the cell detection channel right, I'm going to make a complete guess as to what a suitable threshold would be. You can already run a cell identification of some kind within QuPath, but I think the errors are sufficient that you would really want to come up with something more adapted. But it will enable you to do that, and it will make the measurements in all the different channels currently. So there is a way of representing and extracting the data. In a collaboration with Sandrine Prost, she's working with images, I think, from the Perkin-Elmer vector Polaris system. We had lots of different fields of view, and so when we wanted to maybe begin to look at things spatially as well, I didn't really like trying to solve the problem of integrating analysis information from lots of fields of view. I'd rather solve the problem of, can we stitch them together? And so QuPath will now allow you to be able to take these different fields of view, parse the location, and then stitch them into a single pyramidal OME TIFF image. So you might know about bioformats if you work with images at all, led by a group in Dundee. And so they have come up with an open file format to represent images in pathology, and QuPath then can use their library to write the images, and it can stitch together different fields of view to give you this data. And this was the example I just showed. Was originally supplementary material, I think, with this paper, which I was not involved in, I should point out. And then almost finally, this may or may not work. Nope, my video is not going to run. If you have an image, is my link going to work? Okay. Well, this is going to be risky again. So you might be familiar with the image data resource, again, led from the team in Dundee. If I go in there, so all of these are hosted remotely. You cannot see anything. So these are hosted remotely. So I log in through the web viewer. But what I can also do is I can copy this. I can, I'll close the project, open it, paste it into QuPath. And if all goes well, okay, so now we have it in here and we can begin to run the QuPath is analysis tools or annotation tools if we want to. And so we have access to images, even if they're not there remotely. We can get them from this open source Omero image management system. And so that's going to help with running larger projects at scale. So what I didn't really speak very much about is the interrogating the data, but already within QuPath there's some tools that allow you to calculate a triangulation, so you can begin to look at cell neighbors. There aren't really user-friendly tools to query this at the moment. That's another thing that I think needs to be worked on. But I'm interested in how do you query this data? How do you establish distances from whatever you might want in the most generic way? It can be done partly at the moment and certainly through scripting, but really developing what are the most important commands is going to be a focus of the next year or two. You can download the software, it's open source. This is the community forum. It's the same forum as is used by ImageJ, Fiji, Cell Profiler, and so on. And this is the handbook if you're interested. So when it comes to QuPath, a lot of the features, mostly because they're developed because I needed them. Sometimes they're developed to be nice for somebody else because somebody else needed them, but if I needed them, they've got a higher chance of being in there. But the plan is hopefully to expand the number of developers and to improve this so that it can become a lot more comprehensive in what it does and to make things more user-friendly, to make the deep learning not crash. And so that's what's coming in the future. So some of what I showed you is not, as you can see, ready for widespread use. The best way, if you want to work with it, is to discuss with me as to how that can be done, because yeah, I am currently the bottleneck on quite a few projects, and yet keen to complete these tools so that they can be widely used. So if you want to work with me, let me know. And apart from that, I just want to thank some of the people involved. So this is some of the people I work with at Edinburgh and previously when I was at Queen's, developing in the first case. Yvonne, as I mentioned before, is my partner, the immunologist, and so I need to make her work on her images, and she's put up with me talking about it a lot more than anyone else has and anyone else should. It's an open source project, so it relies on other people's open source libraries as well, like OpenSlide and bioformats to help read the images, OpenCV for some image processing techniques. And so, yeah, it's part of the open source community. And apart from that, thank you for listening. And if you've any more questions, let me know or discuss.
Thank you very much indeed. I have a philosophical question. So I noticed you opened the doc JDK. How are we going to mitigate the harm that Oracle's very aggressive licensing of Java is going to cause to open source tools?
So Adopt OpenJDK also has a Java 8 JDK. One of the benefits, though, one of the small benefits of being the only person really behind this is I could make the brutal decision to switch to Java 11. So now it's at Java 12, and so I've already made that leap. And so I'm kind of away from the Oracle Java.
Yeah, I guess my point is because many biologists are surprised that they've got to install a piece of software called Java on their computer just to run the tools. And now we've got this sort of divergence between what was a standard, which Oracle has been very unpleasant about, and we've got to solve that problem. I mean, it's not a problem for computer scientists or many of the people in this room, but it is a problem for biologists who don't have that background. So clearly you can build it in installers and all those things, but I think there's something fundamental here that we as a field have to do, because more and more universities are going to shut down the use of Oracle on Oracle Java on machines in their institutions. But
my understanding is that you can do pretty much all that you would need to do if you go to OpenJDK, it is missing Java FX, which really affects me here because I use it. But the fact that I moved beyond Java 8 means that that's not a problem either. And so I'm not sure how big a problem it will be as long as you are willing to move to something like OpenJDK as well, which in this case, I am very glad that I did move to the more recent versions of Java. And there's no need to install Java with this. You can package it up to include it with the J package.
Yeah, I'm just seeing a number of tools stopping working or having problems. So it's great that you fixed it, but my question is really about, we've got a wider problem across the field.
I think the wider problem to me seems to be the support for existing tools and how that could be funded and supported. Because it's a problem that I'm facing, and I don't know exactly how this is going to work over the next few years as I try more to apply to get this funded. I think that what something like this and what these kind of open source tools can do is that they can make it possible to do lots of interesting research studies, come up with novel algorithms in a tiny fraction of the time they would otherwise take. But they aren't themselves perceived as being researched necessarily. And I think that is the problem. So these tools that you're describing, I assume they probably could make the leap, but do they have the funding and the support for somebody to help them make the leap? And that is an issue.
I agree.
Hiya.
I would also like to thank you very much for the talk. I am from a genomics background and not from an imaging analysis background. But I do think that the two fields evolve towards each other, and from the genomics background, people would call it spatial transcriptomics, right? And I would like to ask you, could you delineate, is there a difference between what you call multi-channel image analysis and what genomics people call spatial transcriptomics? So, for example, spatial transcriptomics would use techniques like multiplex RNA, FISH. Could you use such a data type for your image analysis algorithms?
I'm happy for others who have worked more with those types of images to answer that as well. So I received some contact about spatial transcriptomic support within QuPath. It's just I've never actually seen or worked directly with the data sets myself. But from as it was described to me, it sounded like it's something that it certainly could be adapted to handle. But it's not something that as I say, a lot of it is developed to the problems that I faced and the data sets that I received, and also had a justification for spending time working on. So I would be interested to know where there are other types of data that this could fit in, and especially if there isn't an existing open source solution that already deals with it, because I don't want to compete with other software, even commercial software. I want to make this maximally useful as opposed to maximize the number of users. And so if there is something where you think it could fit, I would be really happy to discuss and to look at some of the data and see, can we develop the tools in here to make life easier for everybody working with it.
So concerning the spatial transcriptomics aspect, I mean, there's a lot of data being produced in the human cell currently. So, and this is, of course, freely available just as a repository like the one you showed. I think that there is no real standard for spatial transcriptomics so far. It's like single cell 6 years ago. No? So lots of new methods, but no real benchmark. So I think there would be space for applying this to really powerful, already existing image analysis platforms.
Okay, good. It would be good to discuss that some more. Thank you.
Technical question. So you switched from Queen's University to Edinburgh University with the same software. What's the problem? Did you have to ask? Did you have to have an agreement with Queen's University?
It's open source, so I'm using it. I have no advantage in being the person who wrote it in the first place, apart from the fact I know how it works. So yeah, I'm using it under the same open source conditions.
Thank you.
Thank you.